Hello, everyone, and welcome to today's webinar, which is entitled Dirty DNA is OK Sometimes, Obtaining Surefire and Rapid PCR and RT-PCR Results from Diverse Sample Types. My name is Beth Fry, and I'm a product manager here at Lucigen. And before we begin, I just have a few housekeeping notes for you all. First, please note that you, the audience, will be on mute, and you will remain on mute for the remainder of the webinar. But we want to answer any questions that you may have about the material we present today. So we will have some time at the end of the webinar for a Q&A session. And we ask that you use the chat box on the lower left-hand side of your screen to ask your questions, and we'll go ahead and answer them audibly at the end of the webinar. Also, during the webinar, we will have some interactive poll questions for all of you in the audience. This is an opportunity for you to tell us about your experiences with sample prep for DNA and RNA and with PCR, endpoint PCR assays. So the polls are multiple choice, and you will have an opportunity to vote live, and we'll all see the results together. So we hope you enjoy them. We like the polls. We think it's another way for us to interact with you and for you to tell us a little bit more about your research. Hopefully they're a lot more fun too. Another note, if you are watching the webinar in full screen mode, you'll have to exit the full screen mode in order to take part in the polls or to use the chat function to ask questions. So, and with that, I think we're ready to start today's webinar. So our speaker today is Karen Clayman. She is our product manager who handles the PCR products that we're going to talk about today. And Karen has a PhD in bacteriology and many years of cross-disciplinary experience at the bench, including enzymology and assay development for drug discovery. And so we're very excited to have Karen presenting today's subject, which is Dirty DNA is OK sometimes, obtaining surefire and rapid PCR and RT-PCR results from diverse sample types. Take it away, Karen.
Thank you, Beth, and good morning, everyone. Today's webinar is going to focus on two relatively simple topics: nucleic acid extraction and purification, and endpoint PCR assays. What do I mean by endpoint? Well, essentially detecting the final accumulated product after a PCR reaction. And I hope by the end of the presentation I can convince you that dirty DNA is okay for endpoint PCR applications such as genotyping. Okay, so the agenda, we're going to start out with just discussing how endpoint assays are used in today's lab and some of the challenges you may encounter during the PCR, reverse transcription PCR workflows. I'll go through all the various steps and point out some pitfalls in each of those steps and suggestions on how to overcome some of those pitfalls. We'll also talk a little bit about a foolproof PCR system that works for all template types. And then finally, I'm just going to discuss a few examples of quick and effective PCR screening applications, and then followed by the question and answer discussion. Okay, so let's begin. How are endpoint PCR assays used? Well, essentially, they're used for assembly, for example, in cloning, immunogenesis, and sequencing. Next-gen sequencing requires the addition of adapters on the ends of the genomic fragments. That's a great example of assembly. But particularly, PCR assays are also used for detection. And this is where we're going to spend most of our time, if not all of our time, discussing qualitative analysis, yes-no type of analyses. And this is very common for genotyping, looking at SNPs. And also if you're screening a number of different samples and you're just wanting to know if your specific sequence is present or not in that particular sample. Okay, PCR applications have now entered a number of different industries. And again, it's basically confirmation of specific sequences. Now that we have more information coming out of next-gen sequencing and more genomics, genotypes or genomes are being sequenced, more information is coming out. And because when you are doing PCR, you actually do need to know the sequence to design your primers against. So we're seeing more and more coming out of the ag bio industry, looking at seed pedigree determination, the food microbiology industry, looking at authentication of species. Are restaurants really being, are people in restaurants being served the proper type of meat or the salmon versus some other type of fish? So it's authentication. Looking at, in forensic science, human identification and paternity testing and that sort of thing. That's what when it has been used primarily in one of the first applications. And then also in basic research, most recently detection of CRISPR-edited clones. And I'm going to give that example at the end of the presentation. And then finally, in molecular diagnostics in the clinical labs, most of those initial assays were endpoint PCR assays, and a number of them have now moved towards qPCR because the higher sensitivity is required in some of those validated methods. I'm not going to spend too much time on the molecular diagnostic assays and qPCR, obviously. This is going to be mostly focused on endpoint PCR detection. Okay, so just going back here a little bit, this is the general workflow for endpoint PCR, RT-PCR assays. I'm going to go ahead and discuss the various steps, starting with harvest sample, extracting the DNA or an RNA from that sample, discussing a little bit more about what's going on in the PCR reaction. If you have RNA, you do have to convert that to cDNA before you can PCR amplify. And then also discuss some of the different methods and means for detection. But before we do get into that, I would encourage you to participate in our first poll. It gives us an idea of what type of samples you are using, and please just check all that apply.
All right, we've got a number of different sample types listed here, from bacteria and yeast and fungi all the way to mammalian cells and tissue. If you do have another sample type that we haven't mentioned here, please go ahead and type it into the chat window. We'd love to know what you're working with. Looks like there's a lot of folks that are doing culture mammalian cells and tissue. Some people doing blood samples as well. Okay.
Great. It looks like we have a diverse set of samples here. And interesting, the cultured mammalian cells, so maybe that CRISPR application will also be of interest here. So let's move forward with the first step in the workflow for PCR is harvesting the sample. This is actually a very important step in how you collect the sample and how you store it before you actually do the PCR. Because the DNA and RNA can degrade or become contaminated during collection and storage, so it's very important that you process fresh samples quickly and add additives if necessary. Keep away from sunlight. The UV radiation could be bad, and keep it away from heat or extreme conditions. RNA is specifically sensitive to those kind of negative treatments. Avoid cross-contamination. Use sterile swabs. Make sure you label your tubes properly. You do not want to cross-contaminate or mix up your samples. You may need to inactivate nucleases or purify before storage, depending on the sample type and where you may be, and where the PCR analysis will occur. So there's a different suggestions for storing the sample properly, depending on what you have. And all these 4 degrees or frozen at minus 20 or minus 80 is ideal. And sometimes you can go ahead and dry blood samples on filter paper, for example, and then ship those for PCR analysis. Another consideration is that the DNA or RNA yields may be really low or non-representative, and so you have to consider what kind of isolation method you're going to use, and you may need to concentrate the sample, for example. One other possibility is if you're looking for a rare mRNA, you may need, but you don't have much of your initial sample, you may need to amplify, and there are new technologies out there, or some technologies for whole genome amplification that's representative of what's truly in the sample. And Lucigen also now carries a message booster cDNA synthesis kit, which amplifies the messenger RNA in the sample that's representative of the different species. So those are kind of the central things to consider when sample collecting and sample storage before you even begin. And again, if you mistreat the samples, the junk in is going to result in junk out, and you're not going to have reliable PCR results at the end of the day. So once the sample has been harvested and stored properly, the next step in the sequence is extracting the DNA or the RNA. So we'll go ahead and discuss the different options here. So again, the goals of nucleic acid extraction is maximizing the DNA and RNA recovery, maintain the integrity of the nucleic acid, and remove or inactivate PCR inhibitors and nucleases. Sometimes these compounds will come through to the purification process or the extraction process and degrade your RNA or DNA or inhibit your PCR reaction. Some things you also want to consider if you're looking at liquid versus solid or culture cells versus tissues. Maybe you can get by with a cruder prep if you're using pure cultured cells versus a more complex tissue. How many samples are you going to perform endpoint PCR assays on? Are you looking at just a couple, or are you looking at maybe thousands? This can also dictate the type of purification system you're going to use, because you want something that may be more amenable to high throughput processing and automation. And then finally, the amount of sample and the quantity. As I alluded to earlier, you may need to enrich or concentrate the DNA or RNA before you proceed. So these are some general considerations depending on your sample type. Also, each sample type presents its own unique challenges. For example, the cell walls of various organisms are different. Gram-positive bacteria, for example, have thick cell walls, the peptidoglycan, so you need to add lysozyme to break down that cell wall to reveal to the nucleic acid. Yeast and fungi are notorious for having tough cell walls, and so you may need to grind or vortex with beads to kind of break down some of that tough outer cell wall, or to use enzymes. And then plants uniquely have metabolites that are similar to nucleic acids, and so depending on the stage of the plant, you may want to choose to use young seedlings that contain lower amounts of these metabolites so they don't interfere with your nucleic acid through the purification process. Moving on to more complex samples and FFPE samples, I know some of you are using those. You have the added complication of removing the paraffin and the fixatives. The cells or the DNA or RNA is already somewhat damaged in these samples. So you want to be selective in how you really disrupt and remove those nucleic acids from the complex sample. Blood is becoming more and more popular for some, obviously for the molecular diagnostic field, looking at maybe cell-free DNA. So we want to isolate and concentrate white blood cells if you can in some situations, because RBCs, red blood cells, from mammals at least, do not contain nuclei. And you have the added, complication of coagulation with the blood. So that's a little bit more complicated. And then I finally just wanted to mention that there are some reagents that can be used across the board. For example, chitin, the insects and rat tails and nails all contain some similar compounds, and the one reagent can be used readily to extract the DNA or RNA in these samples. Okay, beware of PCR inhibitors in your different sample types. Largely, these can inhibit enzymes. They can, like the DNA polymerase with the reverse transcriptase, they can alter the nucleic acid and make it less amenable to annealing. Some of these compounds can chelate your metals, and the metal is required for enzyme activity. So a number of these have been discovered over the years. In plants, you have polysaccharides and polyphenols that actually affect the nucleic acid and chelate the metals. Melanin in skin and hair can inhibit the enzyme, I think notoriously reverse transcriptase. Indigo that's present in denim jeans and dyes can affect the melting of the DNA and inhibit enzymes. So just some examples of PCR inhibitors that are in various sample types that you may want to consider. And so what are some solutions? What are some strategies to reduce the impact of these PCR inhibitors? Largely, you can remove the inhibitor by precipitation, selective binding, for example, like immunocapture. You can heat treat the sample. This will inactivate maybe some of the destructive nucleases. You can add reagents that bind inhibitors. BSA is kind of notorious for this. Another solution is dilution. Nucleic acid, if you just want to dilute your sample right just prior to PCR, that can be just as effective as well in removing the active component or reducing the amount of inhibitor in there just by dilution. And finally, some of the newer generations of the DNA polymerases have been bioengineered to be more robust and more tolerant to some of these inhibitor, so don't forget that's an option as well. All right, so moving on to the isolation methods. First of all, we want to probably discuss what is the difference really between extraction and purification. And on this diagram, you'll notice the extraction is really just once you harvest the cells and you have a pellet at the bottom of your tube, you can just lyse the cells with a solution and burst open the cells. This is just extracting the DNA from the matrix, the sample matrix. It's a crude prep. It has all the proteins and everything else in there, but it still can be used in many endpoint PCR assays. There are a number of purification methods. The reagent system, which is based on salting out and precipitation and solution-based. You have column system, where the DNA, you remove the protein first, and then you bind the DNA to a matrix, like a silica matrix, and you wash and elute the DNA. This is a good form of concentrating the sample. And then finally, we have the magnetic bead system, which acts very similarly. It's bind, wash, and elute process. Again, another good means for concentrating DNA samples. So all these methods, in the purification methods, provide DNA that's very acceptable and clean for sequencing, cloning, transcription, and expression. I will go in a couple slides discuss this solution-based method and precipitation method, whereby you can use non-toxic solvents in preparing the DNA. But I also want to mention that you can get by, again, just by extraction, just adding an extraction solution and going, heating and going, and it's the nucleic acid is fine enough to go ahead and do some endpoint PCR assays. Okay, so I mentioned salting out. It's an easy and fast method that uses non-toxic reagents. Essentially, you add salt in. The concept is as you increase the ionic strength of the solution, the proteins in your crude extract initially will actually become more soluble because the ions are forming that nice coats around the protein molecules, and so they're more soluble in solution. But as you increase the salt concentration, you actually get a crashing out of the proteins. They find themselves and aggregate, and they precipitate out. So the whole concept is first get rid of the protein by this method, this salting out method, and leaving the DNA then in the solution, in the supernatant after the spin. And then the DNA is then precipitated out of the solution in the presence of salt and alcohol. This is the method that's used in our MasterPure DNA and RNA purification kits. Again, it's just additions and spins only. There's no expensive columns or beads. And you can use a variety of sample types as shown in the left diagram. Basically, you can go add the reagent, lyse the cells, you precipitate by adding a salt solution of sorts, and you spin down the proteins and keep the supernatant. The supernatant then contains the total nucleic acid prep. And then you can split it into two, and DNase treat one and RNase treat the other to get your purified total DNA and total RNA preps. So these are very fast, 30 to 60 minute purification protocols. You get high purity between 1.8, the A260, A280 ratio, 1.8, between 1.8 and 2. These are great for making NGS library preps. You get high yields, they're safe. This method is safe and is very versatile. You can use genomic DNA, total RNA, FFPE RNA, etc. And it's good for both large and small. You can bring down microRNAs for RNA-seq, for example. I also want to mention this method is also pure enough for RT-PCR as well. Okay, so just some examples of high yields that are obtained using the MasterPure kits. This is a figure showing the spin column purification on the left and the MasterPure complete on the right. The lanes represent different amounts of E. coli cells. starting out with 2 times 10 to the 7th in lane 1 and diluting across the gel there. And so you can see that you can detect the PCR amplification product at the lower concentration or the lower amounts of cells in the right panel when you use the MasterPure. So you do get higher yields with this method because you do not lose any on the spin columns or the matrices. It's a very efficient precipitation method. And this is the second figure, actually just shows the different cell types, ranging from mammalian cells to mouse tails to yeast to corn, looking at the different types, DNA and total, nucleic acid and RNA, different preps. So when dirty DNA is not okay, again, when you want to use it for sequencing or for cloning, we recommend the MasterPure DNA and RNA Purification Kit, and we have 5 of these as listed here. And as you'll note, the preparation time is less than an hour, in half an hour in some cases. And again, these can be used for a variety of applications as denoted in the table here. Dirty DNA is okay sometimes. Extracted RNA or extracted DNA is abundant and remains intact for PCR if you do a quick extraction. So as we talked about earlier, you cell harvest and you just add an extraction solution and you heat it to burst open the cells essentially, and these can be used for endpoint PCR screening assays. You maximize DNA recovery this way. This is a quick process, so you can also add some nucleases and proteases in that extraction, and inactivate or diminish PCR inhibitors. So the solution we have at Lucigen are these quick extract solutions. You basically add it and go, heat and go. So heat it at 65 degrees for 6 minutes, and then at 98 degrees for 2 minutes, and then you have PCR ready DNA. So burst open the cells, heat and go. It's very fast, 8-minute extraction protocols for most sample types. Very simple, no centrifugation steps in this particular method. And it's very automation friendly. So you can do 96 well plates, 384 well plates worth of material here. Simple addition and go. So it's very amenable to automation. And again, it only uses non-toxic reagents. And the quick extract solution has gained in popularity for a lot of these screening type of assays, as I will mention later in the presentation. Okay, so just some data, quick extract DNA and RNA purification kits. We here in the first panel show different cell types again. This is using PCR amplification using our FailSafe system. I'll talk to you about this in a couple slides here. But just shows you amplification of different genes here in different cell types, buccal cells from your cheek to hair follicles, mouse tail tissue, bacteria. Again, you get a nice clean band with the extraction and with the FailSafe PCR amplification, the FailSafe kit. Also, this is kind of shows from one organism in different parts of the organism. This is a zebrafish organs, fins and eyes and scales. You can use this quick extract solution readily on these different organ types and get a nice clean band for amplification. And these are a suite of quick extract solutions. We have a couple for DNA, a few for plant, and also for FFPE. And then we have a quick extract RNA extraction kit for RT, reverse transcription PCR applications. And as you can see, a DNA or an RNA that's extracted with this quick extract solution is good for genotyping and genetic studies, GMO testing, SNPs and tumor heterogeneity studies, etc. So here's an example of high-yield RT-PCR products with quick extract and FailSafe. In lanes 1 and 2 is the quick extract RNA extraction solution from Lucigen, and then we have vendor 1 and vendor 2. It's a comparative yield of the RT-PCR products. And as you can see, this is using the FailSafe system with primers for the L-DOA gene, and is separated on a 2% agarose gel. And as you can see, you do get a nice bright band there using the quick extracted RNA, if you will, whereas you don't, and the FailSafe PCR system. And you do not necessarily see that with the other two vendor methods. Okay, that brings us to our next poll question. What methods do you use to isolate DNA or RNA from your samples? Please check all that apply.
Okay. Yeah, so we've got silica columns, magnetic beads, and then solution-based or solution and precipitation-based purification methods. Maybe you're doing an automated extraction method using some laboratory automation. That would be really cool. Or maybe you're working with cells or crude lysates, or we did have a customer that was dealing with virus, so maybe there's no sample preparation that's necessary for those. Maybe you can just go right into your assay. It looks like spin columns is a winner, but there's also a fair number of people that are doing solution-based methods.
Yeah,
right,
thank
we'll
you,
go ahead
Beth.
and stop the poll and continue on. Thank you,
Okay, let's
everyone.
move on to PCR amplification, that step in the workflow. So essentially, once you have your extracted DNA or your RNA, and if you extract RNA, of course, you do need to convert that to cDNA before you can amplify it. And what are the things that are really going on in that PCR tube? And just to point out, the bottom panel is just a schematic of a PCR cycling program, just to remind you of what's going on. The first step is you want to denature the DNA, so you increase the temperature. This denatures the DNA so into the single strands, and then this allows the primers to anneal when the temperature is dropped. Then the primers will anneal, and then they will be extended by the DNA polymerase. And this cycle is repeated several times over, and then you get thousands and thousands of copies of your final product, because it's an exponential production of the fragment. So what's going on in the tube? These are the main components of the reaction. You have your template, the DNA polymerase, primers, DNTPs, and then finally your buffer, which also includes the magnesium chloride, and potentially some additives. So you're really kind of looking at a couple of different factors here, and so I'm going to go and review some of the things you want to think about for each of these steps. So template considerations, again, if you have DNA, you can go straight into your PCR reaction with that, and adding the primers and the polymerase and the buffer reagents, and go. Usually 1 nanogram to 1 microgram of genomic DNA is used per PCR reaction, and that's commonly used for the SNP analysis, is just genomic DNA. And for genotyping, you want to be aware of GC-rich templates, because that may dictate the choice of the polymerases or the additives or buffer conditions that you may want to monitor and alter. And for some cases, depending on the parameter and alter, and for some cases, depending on the polymerase that you're using, you may need to remove inhibitors in your template sample. So those are just some general considerations there. And as I mentioned, if you're starting out with RNA, you do need to convert that to cDNA. On the bottom panel there, you can see this is a two-step process where you first generate the cDNA, and then you can do the PCR amplification by adding the primer and the DNA polymerase, and go ahead and PCR amplify. That's the two-step RT-PCR system. There are also one-step RT-PCRs where you do everything in one tube. You can add the primers, the reverse transcriptase, and the DNA polymerase, everything that's necessary to first make the cDNA and then do the amplification. Usually use specific primers in the one-step RT-PCR. It's high throughput, and you usually can't analyze more than 2 genes in this footprint here. With the two-step, this does allow you to analyze multiple genes, and you can use random primers or specific primers, and you usually can use around the same amount of total RNA. So again, the idea is your template may need to be clean in certain situations. If you have RNA, you have to convert it to cDNA first. And then also take into consideration the GC or AT-rich regions of your template DNA, because that may dictate if you need to add additives or modify your buffer conditions. So moving on, we do have a number of reverse transcription reagents, basically tools for making cDNA. On the left is just a really brief schematic of reverse transcription, and you will start out with the RNA template, and then you can use a number of different primers depending on the application. If you are recovering RNA from eukaryotic cells, you can actually use oligo-DT primers, or you can use random primers or specific primers in generating your cDNA. We have 4 different reverse transcriptases, each having a little bit unique properties, as described on the right panel. And some have H-, RNase H- activity, which also helps in generating the cDNA. Okay, so other reverse transcription reagents, we also have the one-step RT-PCR kit that we offer from Cygnus. It can operate at slightly higher temperatures. We have Message Booster, as I mentioned before. If you're starting out with cells or purified RNA, it amplifies the message RNA in those cases before you convert it to cDNA. And then we have the first strand cDNA synthesis kit as well. Okay, then moving on now to one of the main components of the PCR reaction kit, or PCR reaction, is the DNA polymerase. So what type of DNA polymerases do you really need for endpoint PCR? Well, something that's just really fast, reliable, and really cost-effective. For screening applications, you don't need anything fancy. You're usually just amplifying small fragments for detection. So you just want something that has good processivity, but fast, and these are the ones that it's just your normal standard Taq DNA polymerase. So it's best for the qualitative assays. You may also want to incorporate hot start polymerases. These are polymerases that are inactivated by bound antibodies or chemicals, and then upon heating they are activated. This helps reduce nonspecific primer-dimer, or to reach primer-dimer formation and extension, and allows more more primer sets to be added to the same sample. So you're just improving the specificity by using these hot start polymerases. So these are the main 2 types of polymerases used for endpoint PCR. I'm just going to mention a couple of the others that are used in PCR. High fidelity polymerases. These are generally relatively a little slower because they have proofreading capabilities. These are best used for cloning because you want to maintain the sequence of your template or copy that. You also want to, the other polymerases are also available for generating long amplicons. Some buffer adjustments with some of these, it's usually a blend of high processivity enzymes and with a proofreading enzyme. And so you can get amplicons up greater than 25 kb. But again, for standard endpoint, Taq is just fine. And Lucigen offers EconoTaq for just this purpose. It's a really reliable Taq DNA polymerase and outperforms some of the other vendors out there. This is an example on the left panel here. We have AmpliTaq and EconoTaq on the right. This is just looking at different mutants and different gene products in a sample. And focusing on the bottom right corner, you can see the application products are showing up with the EconoTaq. On the lower left, it's not necessarily showing up with the AmpliTaq, with the CDO mutant. So these are great Taq polymerases for screening. You do have a choice of reaction buffers with or without magnesium chloride, so you can modify that concentration if you wish. This again, it's not a proofreading polymerase, but it doesn't have to be. And we do have samples available if you wish to try it out. Okay, so moving on beyond now the polymerases, the primers are also very important. This is where it all begins. These primers will anneal to your template, and these will then be extended using the DNA polymerase. So when you're designing primers, think specificity and annealing. This is their job, is to find the template, land on it, and stay on it so it can be extended into your PCR product. You don't want any terminal secondary structures in your primers. You want to put a C or G at the 3' end. It's important that you stay, if you can, have a balanced GC content between 40 and 60%. And you want to make sure both primers have a similar melting temperatures between maybe 55 and 65 degrees. Of course, there are now, programs that take all these variables into consideration and can help you design primers for your PCR. And the institute, the companies that you actually get your primers from can also usually have their own programs. So essentially, it makes it a lot easier for you to design primers. But it's good to double check these properties as well, because that could help you with tweaking the reaction. So the reaction buffer can make all the difference. As you may know, it goes hand in hand with the DNA polymerase and the template type. One of those components is the DNTPs. Use HPLC purified stocks and avoid freeze thaws. Again, the higher the molarity, the faster the reaction. That's just kind of basic enzymology, but you do not want to go too high of a concentration. And so you typically use like 40 to 200 micromolar of each. You also want to consider the magnesium chloride concentration, because magnesium acts as a cofactor for the polymerase, and it also interacts with the DNTPs. Again, if you have too much in there, the reaction become nonspecific. So you may, in reducing magnesium chloride and DNTPs to amplify targets specifically and without interruption. So you may want to try that. Finally, the salts and pH. The higher the pH, the easier the template DNA dissociates during heating. So you want to be cognizant of that. And high salt can keep the polymerase on depending on your polymerase. And it can help stabilize the primer binding. Again, you want to make sure that primer anneals and binds well to the template so it can be extended by the polymerase. Okay, so what happens when you have a template that is difficult to amplify? Maybe it's typically you'll encounter this if you have regions that are rich in GC strings here, or the secondary structure of the template is problematic. So you can add additives. Some of these are pretty common: betaine and DMSO, sometimes BSA. A lot of these will help in reducing the secondary structure. It helps with the amplification of the GC-rich regions. BSA can suck up some of the PCR inhibitors and remove it out of the reaction. This is the case like melanin. You can also stabilize the Taq polymerase by introducing maybe some of a little bit of detergent. And the recommended concentrations are identified there in that third column. So you can use this as a reference if you wish. Okay, so what kind of problems or troubles can you encounter during this step? And what if you run your PCR reaction and we've all been there, you see no band on your gel? Well, go ahead and try optimizing the annealing temperature and the buffer conditions. It usually relates to the primer. Maybe it's the incorrect annealing temperature. This is a good idea to try 5 degrees below the lower TM of the primer pair and just do a gradient to see what annealing temperature works best. You want to verify that the primers do not bind themselves or each other. And you may need to optimize the concentration of the primer as well, depending on your sample. Magnesium chloride, as mentioned, can also be improved upon. One possibility, too, is if there's not much of your DNA in your sample, it's there in low quantities, you may need to increase the number of cycles of your PCR program. Another thing you may encounter is that you're going to see multiple bands in your lane and not that nice, tight single band. And why is that? Well, again, your primers may have been designed poorly. You may need to increase the length of them, or the primers do bind themselves or each other, not available, or they bind non-specifically. You may want to increase the annealing temperature. This, forces it to be a little bit more specific on where it binds, and so then instead of the multiple bands, you might zero in on a temperature that where you get that single band that's your gene of interest. Again, one thing you may also want to consider when you do get your primer sets, it's not it's been known to occur that the sequence is wrong. The vendor may have synthesized it incorrectly. So you may want to go back and look at that and have maybe the primers remade. Okay, but a solution to all those optimizations, if you encounter problems with PCR, one solution is the surefire solution of our FailSafe PCR systems. This is a PCR system that Epicenter had first designed and developed and is now being sold through Lucigen. It really avoids the need to optimize PCR reactions. It's never fail. It consists of fail enzyme blend. All you do is add your template and primers to this enzyme blend, and then you dispense that into 12 different 2x premixes, as denoted through A through L in that diagram. In this case, you know, the FailSafe 2x premix J worked best, and I'll show that photo in the next slide. And then you can proceed, but if you wish to purchase just that buffer, you can. It's very easy. And you can proceed then with your PCR. This is a successful PCR the first time and every time. You don't have to think about it. All you do is add it to these premixes and run the gel in parallel, and it'll pop right out for you. This fail-safe system is compatible for multiplexing. And it actually has a higher accuracy than Taq DNA polymerase, a 3-fold lower error rate, and it's extremely sensitive and specific. It works quite well, as shown here. This is amplification of an 80 to 85% GC-rich region of the human fragile X gene with the fail-safe PCR system. So all 12 premixes were tried, the products and were amplified, and as you can see in lanes J, K, and L, you do see the amplification product, the desired amplification product. The brighter band is in lane J, and that's the premix J, and then you just proceed with that. You can also multiplex. This is just showing 5 exons on CFTR from as little as a nanogram of the human genomic DNA, and again using the fail-safe system, and here you're just separating based on size the different exons. When you know Taq DNA will do the trick, we also have an economical form of EconoTaq, of Taq polymerase. It's called EconoTaq. And we have EconoTaq plus 2x master mixes. Just add your primer and template and go. And they work quite well. Ready to use, can be cycled up to 98 degrees. And it's outstanding performance and value for routine PCR. And it fits very well for genotyping and our endpoint assays. Okay, finally, one thing you can also consider is optimizing your instrument settings. This is a reminder of the cycle of PCR. So again, you increase the temperature to denature the DNA. This is usually 94 to 98 degrees. The temperature is dropped, so the primers can now anneal to the template. And then you increase, or in some cases, you increase the temperature to extend the primers. Then this is where the Taq DNA polymerase takes off. It's usually at 1 minute per kilobase with the Taq DNA polymerase. Things you may want to consider at these different steps. Higher temps may be required for GC-rich templates, and therefore you may need to add additives. And if you do, remember you may be able to reduce the temperature if those additives are in there. This is the annealing of the primers. Remember, if you do add the additives, when calculating TM, these additives lowers the TM of the primer template complex. So you have to keep that in mind and may have to modify the annealing temperature. And again, polymerases have different extension rates. The proofreading enzymes have slower extension rates, so you'll have to extend the total time of this step, so to make sure that you get your full product. And then finally, you don't want to go beyond beyond 40 cycles. At this point, most of your reagents are already depleted, and you're going to get more non-specific amplification and that sort of thing. Okay, so we're going to move to our third poll. When your PCR reaction fails, what do you do?
All right. I don't know about you, Karen, but I think this is a situation where most people have found themselves in at one point or another. No bands on the gel. What do you do first? Do you optimize the cycling conditions using the same polymerase you've already used? Maybe you optimize the reaction formulations, like maybe you try enhancers like betaine or DMSO. Or do you evaluate other polymerases? Do you just switch and use a different enzyme? Or other PCR master mixes that don't allow you as much optimization? Or maybe there's something else that you do. If there's something else that you do as a first triage, please type it in the chat box. We'd like to know what it is. All right, looks like most people will use the same polymerase and just optimize the cycling conditions.
Yes, thank you.
Thank you, everyone. Moving on.
Okay, let's move on to the final step in the workflow for the endpoint PCR is really analyzing the results. How do you know you are successful? So you can separate your product based on size or fluorescent readout. There's a way to detect the product by fluorescence measurements, or both, based on size and fluorescence. So it gives you a variety of different options. So you do have to consider, your application. Do you just need a quick, dirty method in a research lab, like you're screening clones? Or are you in a situation where it's a validated method in the clinic, and it's very important that you have the sensitivity required, the equipment that can sense the sensitivity, lower limits of detection, for example. Are you limited to gels, or do you have some of these higher end pieces of equipment? Those are something you can think about. Again, how many samples will be processed at one time? You need the equipment to accommodate these the high throughput of all these samples. You don't want this to be your rate limiting step. And then one other thing to consider is your post-PCR handling. Once the PCR reaction is over, there's a lot of your amplicon in there, and it's important not to open the tubes because there's potential to contaminate your environment, which would contaminate any future samples in that area. So sometimes it's advised if you can use a system where that tube will not be opened again after the PCR amplification reaction is completed. That's also something to consider for your application. So again, you can use gel electrophoresis, separation on size, or capillary electrophoresis. This is size and fluorescence detection. Or else you can use your normal plate reader that's in the lab for 96 or 384 well situations, or the qPCR assay can also be used because you can do your PCR reaction and your detection at really the same time. And this is just a chart showing some of the pros and cons of each of the different systems. Just basically, again, ranging from your gel, which is very cost effective. The gel apparatus is very common in most laboratories and is easy to use, all the way up to qPCR thermocyclers or high-end plate readers, which obviously can allow for more high throughput and maybe even time savings because you can just put your reaction in and walk away. Okay, so that brings us to the first example of how you can use endpoint assays in industry and in the research lab. And the first example I'm going to discuss here is the CASP genotyping assay for SNP detection by LGC. This is a homogeneous FRET-based assay, which consists of the CASP assay mix. This consists of two different allele-specific competing primers with unique tail sequences. Okay, and just to reiterate, the CASP actually stands for Competitive Allele Specific PCR. So you have two primers in there that are competing for each other for the template. And then you also have a one reverse primer, which can be used for either one of these. The kit also comes with a CASP master mix containing the Taq polymerase and the reporter FRET cassettes. So in their inactive form, as this is they're quenched, you do not see any fluorescence. Once they're used for reporting, you will see an increase in fluorescence because these will denature and the fret will be released or interrupted, and you'll get fluorescence. And then finally, you do get a test DNA with the SNP of interest in the kit as well. So the method is basically you denature the template and anneal the components, and the two primers with the different allelic specificities will compete for the template. And only one of the primers will bind properly and will outcompete the other primer because it hybridizes much better to the template because it's a direct match. These primers also have a reporter region, and this is exactly where the fret cassettes will come in handy after multiple PCR reactions. You'll get extension of that reporter sequence, and then the reporter fragment from the fret base will now bind to the complement, and now you'll see a fluorescence readout because it's no longer quenched when it's attached or annealed to its partner over here. And so these are hex and FAM dyes, depending on which primer is annealing to the template, and you can get different colors representing heterozygous or homozygous readouts. So in this case, the genotyping marked as red are homozygous. For the allele reported with hex. Those marked blue are homozygous for the FAM allele, and then those that are marked green are heterozygous. So you get this kind of a pattern, you can normalize it, and you can get this fret-based readout using this very competitive allele-specific PCR genotyping assay. It's amenable to high throughput, and it's an easy readout. And this is one example where the researchers here in Australia use this SNP assay for looking at various variants of wheat. And what they were looking at was different SNPs that were related to certain traits. For example, higher yield or resistance to crops or disease, and also just the higher quality of the yeast or the wheat itself. And so essentially they were looking at different variants from different countries. The diagram below shows 4 different colors representing 4 different countries, and they're just basically looking at the frequency of these unique traits in the different samples that were collected from the different countries. And by looking at, and there's more than 8,000 pre-validated assays that are available for breeding in wheat. So you can see the power of this when you're looking to potentially breed for crop development in the future. And these are all publicly available markers and can be readily ordered here through the LGC. So very powerful for, and as we get more and more information and more genotypes are sequenced, we'll identify more unique traits, and we can zero in then, on crops that will be more improved than in the past. I also want to mention one of the more common methods of using genotype assays. These are screening assays for mutant or wild type mice. In this example, the researchers were looking for the insertion or the disruption of the PDS5B gene with the HER2 gene. And basically, you can just take the rat or the mouse clippings and treat them with QuickExtract and run your PCR reaction, and you can look at the different patterns on an agarose gel as shown here. So this is wild type. The presence of the PDS5B gene is evident. If one of the copies is disrupted, you'll see 2 bands. And if both copies of the PDS5B gene have been disrupted by a HER2, you'll see only the bands that pertain to the HER2 gene. So this is a very common method application for genotyping or endpoint PCR reactions. And then finally, I want to talk about identification of CRISPR-edited cell pools or clones. These are many common methods require target site amplification. There's many methods for looking at this, and I want to focus on the last one, the endonuclease mismatch cleavage or EMC assays. or also referred to as the T7E1 assays. It detects indel mutations quite well. And so the workflow for the T7E1 assay is depicted here. You lyse the cells and you perform PCR. And PCR amplicons containing mutated and wild-type sequences are then heated up and then allowed to slowly cool down. And then, so you'll have heteroduplexes where the mutation has occurred, where the matching on the right, for example, where the T does not match with the C, it'll still form that heteroduplex, but the T7E1 endonuclease will recognize that mismatch and cleave it. Whereby if you have the homoduplexes, you'll see that you get non-cleavage with the T7E1 enzyme. And then all this is analyzed by analysis on an agarose gel. I want to point out the challenge is the PCR. You want to make sure you have one band and not a smeary product. You don't want non-specific amplification because that could impact the results of your T7E1 analysis. QuickExtract has been highly recommended and cited for T7E1 assay sample preps. It's very efficient lysis and extraction. It's a fast protocol, and it is scalable. You can look at one sample or up to thousands at one time, and it's compatible with many high-fidelity polymerases. So just briefly, it's again high throughput. You can go ahead and pipette your cells into a 96-well plate. Add QuickExtract, heat it quickly for 6 minutes, 98 degrees for 2, and you have PCR-ready DNA. Then you can amplify with Q5 polymerase. Look at the PCR products on a gel. Make sure you got a single band and it looks pretty uniform. These are different clones or pools. And then you can go ahead and form that heteroduplex formation and digest with the T7E1. And these are the results. These actually show positive editing results. We don't have a negative control in here. You actually do see the digestion products showing that the heteroduplex was formed. So QuickExtract works extremely well for these type of assays. Okay, we'll go ahead and just go ahead with this final poll. Just briefly, do you plan to use endpoint PCR assays for screening applications in the next 6 months? And how would you characterize your lab? And then we're almost finished with the presentation.
Okay, so this is kind of 2 questions in one, right? So everyone can make a yes or no choice. Do you plan on doing this in the next 6 months or not? And then, please just let us know if you can what kind of lab you would characterize yourself as. Maybe your basic research, forensics, clinical, food microlab, bio, or if there's something else that we haven't listed here, please let us know and type it in the chat window. Looks like a lot of basic research, and people are doing PCR for screening applications in general. Kind of a mix there. All right, great. Thank you, everyone.
Great. We'll go ahead and then summarize. Basically, we've discussed how endpoint PCR assays are alive and well in today's lab and can be used for many types of SNP analysis and transgenic animal analysis, and more recently with CRISPR-edited clone identification. The QuickExtract extraction kits provide a really easy, simple method that can be used in high throughput processing, and it can be used for a number of different cell types. It's amendable for PCR, RT-PCR, and I also wanted to mention these quick-extracted DNA can be used in qPCR assays as well. And I want to, finally, the FailSafe PCR system was discussed. It's a real good system, just ensures one-time surefire success in getting your amplification product. And with that, I just wanted to mention these are the components or products that were mentioned in today's webinar. They're available here for future reference. And with that, we'll take some questions.
Okay, great. Thank you, Karen. If you do have questions, please use the chat box in the lower left-hand corner of your screen to type in your questions. We'll be happy to answer them. We did have a question during the webinar. Beth D, who, lovely name and lovely question, she asked if we can use the MasterPure gram-positive DNA kit for qPCR as well. Karen, you have any thoughts?
Yeah, yes, absolutely. We do have, that's, with the MasterPure, you can get very pure DNA for a number of different applications, and yes, qPCR is one of them.
Okay, do we have any other questions from anyone? All right, well, I guess I had one on behalf of our audience. If people are working with various kinds of samples, so everything from virus to tissues to cultured cells to bacteria to gram-positive and gram-negative bacteria, if people have a question about which kind of either extraction or purification kits to use, how would they go about navigating that product line?
Yes, at this point we do have like 4 QuickExtract products and 5 MasterPure, and we probably, in most situations, have a solution for you. I would recommend at this point, we are assembling different, sample types and how they can be, how the nucleic acid can be extracted or purified with any of the kits. But in the time being, I would go ahead and just contact our Lucigen tech support at lucigen.com, or give us a call, and we'd be happy to guide you through that.
Okay, great. Well, in the interest of everyone's time here, I think we'll go ahead and close the webinar. We'd like to thank everyone for joining us. We will make both the webinar recording and the slides available on our website in a few days, and you'll be receiving an email when those materials are available, so keep an eye out for that. And thank you very much for joining us. Thank you, Karen, for a great presentation, and thanks to every member of our audience. We appreciate you joining us today.